Welcome to The Explainer from BioinfoBytes. Today, we're cracking the ultimate biological code, DNA fingerprinting. Think about it, identifying a single person out of literally billions using just a tiny microscopic trace. It's totally not just TV magic, it's hard science. So let's get into it. Picture a totally cleared crime scene. The yellow tape is up, and to the naked eye, well, absolutely nothing is left. But hidden deep in the carpet fibers, maybe there's a single hair follicle, a tiny drop of sweat, or even just a rogue skin cell. To a forensic scientist, that invisible speck isn't nothing. It's an entire biological manuscript just waiting to be read. Which really brings us to our central mystery today. How exactly does a microscopic trace reveal a totally unique identity? I mean, how do we actually go from a tiny degraded fragment of biological material to a definitive, undeniable confirmation? The genetic blueprint. Okay, let's dive into this. We always hear that DNA is the blueprint of life, right? But here's the kicker. Forensic scientists actually don't care at all about the 2% that codes for traits like your hair color. Nope, they focus entirely on the other 98%, the non-coding region. And their big secret? Short tandem repeats, or STRs for short. You can kind of think of these as your own unique genetic barcode. See, within our DNA, short sequences of base pairs just repeat over and over again. And the absolutely crucial part is that the specific number of times they repeat varies wildly from person to person. Now, what's really interesting about this slide is if we look at a specific STR marker, like THD01 here, you get one copy from your mom and one from your dad. The top sequence might repeat 7 times, while the bottom one repeats 8 times. And that exact 7 and 8 combination, that is a literal piece of your completely unique biological signature. The laboratory gauntlet, because spotting the DNA is one thing, but actually extracting it, that is a highly rigorous, fast-paced technological gauntlet. Here's how that magic happens in 3 rapid-fire steps. First up, extraction. A centrifuge spins and concentrates the sample. Second is amplification. This is where multiplex PCR essentially photocopies specific STR regions millions of times. And finally, separation, where a process called capillary electrophoresis sorts those tiny DNA fragments absolutely precisely by their size. Reading the DNA profile, this right here is where all that complex chemistry gets converted into a clean digital output that we can actually interpret. The machine spits out what's called an electrophoregram. You can think of this chart as the ultimate translator of our biological barcode. It displays the genetic material at each specific location being tested, and every single peak represents an allele, which perfectly corresponds to those repeating sequences we talked about earlier. If you take a look at this single source profile, see all those really sharp peaks? Each of those gray bars corresponds directly to a specific STR locus. It's kind of like a jagged mountain range of data, and it is the literal digital manifestation of a single individual's genetic identity. Matching and CODIS. Okay, now that we have the profile in hand, we need to zoom out to the societal level. This is exactly where the hard science meets the criminal justice system, specifically inside the FBI's massive database. And here in the United States, this whole system revolves around one absolutely critical number, 13. For a profile to even be uploaded to the National DNA Index system, it must test a minimum of 13 specific core STR markers. It is an incredibly rigorous, highly calculated standard, and it's designed for one thing: absolute certainty. So the crucial point is this: a match at all 13 loci? The probability of 2 unrelated people sharing that exact same profile is less than 1 in 10 billion. So unless you have an identical twin walking around, that profile is yours and yours alone. But if there is a mismatch at even just one locus, boom, the suspect is 100% excluded. It's a definitive biological alibi beyond the crime scene. Because this powerful technology isn't just used for catching perpetrators. It brings huge amounts of closure through disaster victim identification, it exonerates the wrongfully convicted, and it is pretty much the absolute gold standard in paternity testing. And this brilliantly illustrates exactly how family testing actually works in practice. On the left side here, the child has a 6 and a 7 peak. We know the 6 came from mom, so that 7 absolutely must have come from dad. And look at that, the alleged father has a 7, so it's a match. But on the right side, the child has a 29 and a 31.2. Mom gave the 29, but the alleged father is totally lacking the 31.2 peak. That's a mismatch. He is 100% excluded as the biological father. So, from a completely microscopic hair follicle all the way to a jagged digital chart of peaks and valleys, it all really leads us to one undeniable truth. Your STR profile is uniquely yours. It's an irrefutable barcode that's literally written right into your biology. But as forensic science pushes even deeper into the molecular level, you kind of have to wonder, what other hidden biological secrets might the invisible traces you leave behind every single day soon reveal? Thanks for joining us for this explainer from BioinfoBytes.